Welcome, everyone. I hope you can hear me on Zoom. Welcome to the joint seminar of the Precision Medicine Resource, Columbia Precision Medicine Initiative, and Center for Precision Medicine and Genomics. We have these joint seminars now that are every 2 weeks, and I'm very excited to introduce our speaker today, who is Tsang. He's a tenured professor of pathology and cell biology and ophthalmology here at Columbia. He's an MD PhD. He completed his PhD in 1996 in stem cell genome engineering. He also went to Columbia BPNS and graduated in 1998, then went on to complete a fellowship in ophthalmology, specifically in electroretinography at UCLA. In 2003, he came back to Columbia, where he built a very successful NIH-sponsored research team and continues to do really exciting research on a variety of different topics related to inherited retinal diseases. He has been involved in the development of preclinical models of inherited retinopathies. This involved testing genome engineering strategies and preclinical models, also developing patient-specific models of disease with knock-ins and iPSC studies. And he's developing gene and stem cell-based therapies for retinal degeneration. He also has a very vibrant clinical practice where he sees a lot of patients with very rare inherited forms of retinal disease. And he's applying both kind of precision medicine diagnostic and treatment approaches. So welcome. And we're happy to have you today.
So thanks, Christoph, for having me to share you some of the ongoing work. And so I just to show you, to start with, this is application of precision medicine that you pay for it. Medicare, Medicaid since 2017 pays for gene therapy. Gene therapy maybe is one of the ultimate application for precision medicine. And I'm especially grateful for you in the room because the same time across the street, Nobel Prize lecturer from protein hydroxylase just across the street. But I'll tell you a little bit about my version of the protein hydroxylase also. So this is it is something that Lisa Hack here, the director of clinical trials here, also together Tongue Up the Cell do here. So this is show you injecting DNA into a patient. This is what Medicare approval for the first gene therapy, the gene called RP65. So you can see that the gene is injected. This is the optic nerve that connects the eye to the brain. This is the macula. So this is a bubble injection of the DNA. So this is a pre-op before surgery. This is actually a download from New England Journal of Medicine, and this is the data that was presented to FDA, leading to the first gene therapy approvals. So this is the patient has a camera on his head, and his task is to walk from this side of the room to the other side. So this is a patient. This is pre-op before gene therapy. And so the primary outcome measurement for FDA is to check the time it takes to go from one side of the room to the other side of the room, and also check how many times a patient bump into the wall. Sometimes patient get, you need a guide because sometimes patient get confused and walk to the wrong direction because the FDA trial is to go from one side of the room to the other side, not walking backwards. So this is a pre-op before surgery. So it is a camera you can see on his head, so you know about what the patient is looking. And then the next video will be post-op. Usually in the efficacy peaks around between 6 months to 1 year after surgery. Move a little faster. So it takes about 77 seconds to go before pre-op, before gene therapy. And now this is a patient post-op. Same patient. the same patient. It's about 6 months after surgery, injecting the gene. Show you the same patient. So it was 77 seconds to go from one side of the room to the other side. And this is actually quite typical result for this particular Medicare, Medicaid approval. So it's about 14 seconds as opposed to 77 seconds, and there's no bump. So now I quit this one and then maybe share the PowerPoint. Let's see it works, hopefully. Let's see. Stop sharing.
You will share this screen, right?
Yeah, share the screen, the new one.
We'll just start sharing. And so like that's
Maybe this one.
Yeah.
Okay, hopefully it works for the Zoom audience.
Yes, it does.
Okay, great. Thank you. It's coming out. So we are the College of Physicians and Surgeons. So I've got some of the concepts to tell you about precision medicine. That medicine and surgery is not the same.
Just sorry, we see it as a presenter mode when we can also see the next slide.
So so surgeons and medicine used to be a different culture until the formation of the College of Physicians and Surgeons. So you can think of genome surgery and gene medicine also different. Surgeons don't go to school, they go to barbershop. And it is a formation, and internists, medicine need to go to school. You need to look at the urine glass to look at different color. And then not too long ago, I remember some of the senior attending when I was in medical school here, they hate going to the diabetes clinic because I think the resident's job is to taste the urine. So you need to, so medicine and surgery has a different culture until the formation of the College of Physicians and Surgeons. And surgeons also go to school. So surgeons now go also come to school after the College of Physicians and Surgeons. So the culture is different. Which is here, try again.
It was fixed after you changed it.
So you can see can you see the
I wouldn't see anything anymore, nothing is shared.
Okay, share, try again. You saw the logo for PNS?
Now it's perfect. We can see the PNS and yeah.
Oh, this is a logo for PNS with the laser.
Surgery, yeah.
Thank you.
So we take some example of how genome surgery is in the context of precision medicine. So we start from the most precise. As we go down, they'll be less and less precise. We tell you about precision repair of the base pair, each base pair at a time. And then we want to see how can treatment is so precise, but then, for example, for monogenic disorder, there's 150 mutations in the gene rhodopsin. So how can we treat all 150 mutations? It's challenging to go to FDA 150 times. And then we talk about then maybe even less precise, maybe looking at the pathway instead of the gene. So it's not uncommon in the world of genome-wide association that we get we're not sure what is causative. So in the field in the last 20 years ago, one of the best risk factor for macular degeneration is in the 10q locus. But it's not clear whether or not ARMS2 or HTRA1 within the 10q locus, because they are linked. You cannot use families to segregate them. So it's been debated many different ways whether or not ARMS2 or HTRA1 causes the disease. So we started it actually BC, the project for the fellow Lulu started BC before CRISPR with talent, and then we also supplemented with CRISPR to change one by one. So in the ARMS2 allele, there's two risk allele. There's a G allele that is low risk, but then if you make it into the T allele, then then there's a 70% chance that the patient would get macular degeneration. And also it's been debated in the HTRA1 is a protease, and Genentech has tried to inhibit this protease, but to see whether or not it's efficacious in macular degeneration. And then the low risk allele is G, but the high risk allele is A. And also between the arms and HTRA1, there's an insertion deletion called 443. And then in the risk allele become 54 residue. So it's not clear because in this 10q allele, this is linked, which one causes the problem. So with genome surgery, that we just change one by one. So we change it to the low risk allele G into a high risk allele T without affecting HTRA1. And then also we did the same with HTRA1 and then changed it from a G into a high risk A without affecting the other ones. Then we take these patients' stem cell and then turn them into retinal cells. And then assess the outcome measurement we use is looking at oxidative stress, partly because on untargeted proteomics that we saw that the low risk patients have very high levels of oxidative defense, in particular the gene superoxide dismutase. So this is macular degeneration. If anybody know what painting this is. The owner for this painting actually has retinitis pigmentosa instead of macular degeneration. So doesn't see the side. The owner for the painting make a bump at the elbow right here. And so the painting has changed the value after the tear from $160 million to $80 million for this painting. But anyway, in macular degeneration, you don't see well in the center, as this is some of autofluorescent imaging developed by Janet Sparrow here that are looking at vitamin A metabolism. So then we took the high risk, the low risk patient cell and do one by one make them high risk. And then these are retinal cells that can make from patients' stem cells. Over time, that the name of the cell is retinal pigmented epithelium. So over time, that is become more pigmented over time, as the name say. And then so we have both low risk cells and also the each of the edited cell as isogenic control. So in the high risk cells, the patients have very low level of superoxide dismutase activity. In the patient, they don't have macular degeneration, the low risk allele, they have very high level of superoxide dismutase. When we do the editing to just change arms 2, then the trend, it converted the low risk cells into the trend of the high risk cells. Versus if we do the editing in HTRA1, it seems to change less. And we assayed it by just growing the cells over time in culture. And also we artificially aged them with peroxide and also using one of Professor Janet Sparrow protocol to add this retinoid. So you can, because we do some pharmacological aging, because none of the graduate students want to grow the cells for 60 years because macular degeneration don't manifest in 60 years. So you can see that it's the same in pharmacological aging, that the low risk cells have very high level of superoxide dismutase activity. The patient with macular degeneration high risk have low level. And then when you do the editing of arms 2 rather than say HTRA1, we can see a better effect on the superoxide defense system. And looking at cell viability, in low risk cells, of course, they grow well, they don't die. And then other over culture of 16 weeks or other exposure of Professor Janet Sparrow's bisretinoid. And then you can see that the low risk cells, they survive pretty well, even you age them pharmacologically. And then in the high risk cells, they die. And then a single edit of arms 2 lead to the same fate into high risk cells, versus the editing in the HTRA1, it doesn't seem to do too much to change the viability in pharmacological aging. So this just to show the summary. So a single edit, we will argue that we have solved the controversy whether or not in the 10q locus, whether ARMS2 or HTRA1 causes the risk for macular degeneration. We'll argue that because a single edit of these low risk cells of ARMS2 at the 69 residue, that will make it features of the high risk cells, versus the editing on the other two, HTRA1, although this is being drugged by Genentech, editing at the HTRA1 does not seem to contribute to the outcome of oxidative stress, superoxide dismutase activity, or cell death. So we argue that in the 10q locus, in this genome-wide association study, ARMS2 will be responsible for all the genetic risks. And also in treatment response. So this is antioxidants, PBA. This is the only approval for Lou Gehrig's disease. So Lou Gehrig, the last 20 years, only approval is this antioxidant called PBA. And then of course in low risk patient cells, you give them PBA, they do pretty well. They don't have very low level of reactive oxygen species, have pretty good viability in low risk cells. When you expose them to Janus Sparrow's bisretinoid pharmacological aging, the high risk cells, of course, they die. But then you give them PBA, antioxidant, then they survive much better. Blue light also in artificial aging. The single edit of the ARMS2 will now make the low risk cells behave like the high risk cells in terms of responding to a drug, PBA, versus the editing in HTRA1 also does not seem to have any much effect on this response to PBA. So this is another, I guess, precision pharmacological response to treatment. It's also dependent on the genotype. In our case, we also argue the response in the 10q locus is to PBA is all due to ARMS2, not so much in HTRA1. So this is, I think this precision editing helps us to solve some of these genome-wide association locus. This is not as robust as some of the assays we do in tissue culture for monogenic disorder, but I think this is typical in other fields. In Alzheimer's, looking at GWAS allele, they tend to be effect a little bit weak. We apply pharmacological aging for this age-related condition because graduate students just don't want to grow cells for 60 years. And then we also show the response to treatment instead of specific to ARMS2. So in this 10q locus, we argue that most of the effect is due to the risk allele for ARMS2 instead of HTRA1. So now this is pretty precise, and then we want to see applied precision genome surgery to treat many different mutations in this dominant disorder. So recessive. So most of the FDA trial in recessive disorder. So recessive. So usually for people, you have two happy protein, one from mom and one from dad. And a recessive disorder that most of the current trial is about. Then you have a sick protein, you give them back a healthy cDNA, then you get back pretty normal. In dominant disorder, so we like to focus on dominant disorder because most of the pharmaceutical currently are focusing on recessive disorder. In dominant disorder, you get one good protein and one crazy protein. And the crazy protein destroy the dominant negatively, the good part of protein. So you do the conventional using gene as a drug, then you also get destroyed. So really for dominant disorder, you need to cut it off, cut out the crazy one, and then get back a good one, either repair or just cut it out. So we have done so conventionally, as I show you in the macular degeneration GURs, that you can use repair specifically. They are very cleanly scarless genetic surgery, mutation by mutation. We don't have time to go through today because mostly it's published already. We also one approach in dominant disorder is converted into recessive. So we can successfully ablate both the wild type and also the mutant allele, and then replace it with a CRISPR-resistant cDNA. So changing a dominant disorder into a recessive condition. But we'll focus for today is another strategy is to kind of use single nucleotide variant to map the disease versus the wild type chromosome, and then specifically ablate the entire anything linked to the single type, the mutant linked to the single type nucleotide variant. So most of our work is all inspired by patients. So this patient come in and never thought of working on rhodopsin, but the patient's family looked pretty interesting. So this is a optical imaging. So this is the actually the unaffected child, and the proband is this 51-year-old. You can see that the photoreceptor nuclei, just like in the mice. So we eventually make a mouse model that matches the patient mutation. This is a slow degeneration. This is the nuclei of the light-sensing neuron, the photoreceptors. And then so you can see that some problem with biosynthesis. So the outer segment where rhodopsin lives, the membrane a little bit short. And then over time, it's the nuclei of the light-sensing neuron also goes away. These little dots here. And then in the patient, you can see that also this the correspond is the nuclei of the light-sensing neuron also goes away. And then in the 16-year-old is not so much affected. And the 12-year-old is not so affected, although carries the mutation. This is a slow degeneration, just like the mice. And this is the unaffected sibling control. So we have treated this mutation called D190N and also another more common mutation, protein 23 his. And this ablate and replace, converting a dominant into recessive disorder. So you can see that the rescue cDNA, CRISPR-resistant cDNA, still lasts a long time, even after one and a half years, 500 days in a mouse. This is a slow degeneration. At 500 days, it still a single row of nuclei. And the treated one in 500 days still has more outer nuclear layer, more cells are surviving. So this ablate and replace strategy works. So we work on many different mutations already, at least 3 we show. So now we're going to show you some of the new ongoing unpublished data on looking at taking advantage of single nucleotide variant to map the entire chromosome. So the idea is that the mutation is linked. If the mutation is linked to the single nucleotide variant, as shown in here, that's too much. So if it's linked to the mutation, that we can use one guide RNA directed at a single type nucleotide variant, and all the mutations in between. So we need an upstream as polymorphism. And then any mutations in between that we can, together with a second guide RNA, we can pop out everything in between. So for dominant negative disorder, it will spare, CRISPR is so sequence specific, it will spare the wild type chromosome. So we can now, for true dominant disorder, if there's no haploinsufficiency, so that this should work. So we can specifically ablate anything that are linked to the single nucleotide variant, and then spare the other chromosome. That's the idea. So then to show you that, say, we can specifically target the G or the T allele, and if it's along, and if the mutation right in the middle is linked to the mutation, we can ablate the chromosome. But then if we can use a different guide RNA to recognize, say, a T or a G different from the wild type chromosome and target the other chromosome. And of course, in this strategy, if patients are homozygous for the SNP, it will not work. And it is just looking at the patient collection with different rhodopsin mutations. And so about in the general population, about 40% are heterozygous. So we can use this SNP editing strategy to cover about 40% of the patient. This is just to show you some how it works. That we can also, to increase the specificity of the targeting the SNP, we can also create some additional mutation in the guide RNA. So then this is a residue that would target in the SNP, and then we can create guide RNA homology to the A. And so in this say 13 versus here, this is a parental band, and then specifically we cut the A, and then if it's a G, the cutting efficiency is not as good. So if this happen to be the template is the patient template to be G, and this guide RNA targeting the A, but will not touch the G template if they for the patient. So this is just another illustration. So we can they are looking at the SNP at the PAM site. So you can see that the guide RNA 13 would target the A, would cut the A template. So the A template, this is coming from the patient. If the patient happens to have a G template or the other chromosome, the guide 13 will not cut the G template. Switching another guide, the guide 14, supposed to target G. The guide RNA targeting G 14 will not cut the A template, versus the guide RNA targeting G would able to cut the G template. So we can pretty specifically using single nucleotide variant to map the paternal or the maternal chromosome that could be linked to the mutation. So CRISPR is sequence specific with the help with the support of the Precision Medicine Initiative from the Roy Vagelos Fund. We generated mice that carry the human SNP, carrying human sequences, because otherwise CRISPR is sequence specific, will not target mouse rhodopsin. So then the one we made that you can see that over time, just to show that it works, it degenerate. So we also need to generate the human wild type rhodopsin as a pair also. So then the rod specific response, the night seeing cells a little bit down compared to the wild type control. And then the cone responses, because this rhodopsin is rod specific, the cone's response at this stage is fairly spared. But you can see the rod response went down in this future recipient for CRISPR therapeutics, so this humanized rhodopsin mice at the 347 residue. So you can see that in the wild type, so the human rhodopsin works in mice. So we replace the mouse with the human rhodopsin. In the wild type cases, the human rhodopsin, just like the mice, the cells don't degenerate. And then the one carrying the dominant human mutation in one chromosome, then you can see that the light sensing neuron slowly degenerate as in the cursor, also show on the plot. So this seems to mimic human condition. This is the lunar new year of the rabbit. In ophthalmology, rabbit count as a large species because FDA submission typically require non-human primate. But then we also generate rabbit. We hope the FDA will accept rabbit as an alternative to monkeys. So we have also a different mutation. We have rabbit. This is an optical imaging. You can see that rabbit, the control, the nuclei looks pretty good. And the electric response in the control is pretty robust. And then in the rabbit, electric response, the retina also extinguished compared to control. And then you can see the nuclei in the rabbit also have less than the control. So in the lunar new year of the rabbit, we also have now rabbit carry human rhodopsin sequence to test CRISPR therapeutics. So we hope that with just 2 guides instead of 150 guides, we can cover at least 40% of the patients with rhodopsin mutation. So in the remaining time, so not uncommonly that maybe even with whole genome sequencing, 20% of our patients, we cannot find a gene in 20% of the patients. So what can we do to treat those patients that when we clearly it's monogenic, we just cannot identify the gene even with Megan's help on our whole genome sequencing. So we think maybe there's some common pathway. Maybe we can treat the pathway, although maybe not as effective. And this is inspired by patients that come to see me. So there's a patient referred for electrodiagnostic testing with poor vision, can only count fingers. Recently noticed night blindness. One eye is still pretty good vision, 20/40. In fact, it's a hematology is a hematology technician that counts cells in Brooklyn in downstate. This is from this picture. You can see the large choroidal vessel looks pretty well because it's called window defect. Epithelium is not there in this patient. And this is his visual field. And then on the electric response, you can see that typically has patient a pretty good control, has pretty good rod-specific responses. And in the patient, there's no rod responses. And the cones showing later that it spikes in cones. I show you this patient and a couple of others with the same diagnosis. And then you can see that the cones are relatively preserved in this condition. This is looking at one of the Janet Sparrow's bisretinoid imaging. You can see that when the cells go away, you can see that the epithelium go away. You can see the choroidal vessels pretty well. Optical sections, you can see that most of the nuclei disappear. That's why he doesn't see so well. And the epithelium is pretty thinned out. You can see there's not much epithelium. And you can see the choroidal vessel very well. This is a live imaging because there's no epithelium there. Typically covers the choroid. Otherwise, in here is relatively normal. You don't see the vessels very well. So this is a milder patient. This patient was at an earlier stage. This is not so uncommon. In fact, it's more common in the internal medicine clinic than coming to see us. So anybody can recognize the diagnosis based on the facial features. So this patient is very interesting. They're making bone marrow from the head, and this patient. Any suggestion what the diagnosis? So this patient has thalassemia. So it's taking iron chelator. So now it brings us to you missed the Nobel Prize lecture because the protein hydroxylase requires iron to work. The Nobel Prize winning enzyme requires iron to work. So we take iron chelator, so the Nobel Prize winning enzyme will not work. So they take deferoxamine because of transfusion of thalassemia, need the iron chelator. So you can see this patient, and very interesting that it takes the iron chelator, but the most sensitive cell is actually this epithelium. Epithelium support the photoreceptor. In the last talk, I'm mostly telling you about photoreceptors, but now the epithelium actually support the photoreceptor. And this is the relationship, the epithelium here, and this is a photoreceptor. And this is like a cilia. So photoreceptor like a cilia, and that's why a lot of patients with Joubert syndrome, the ciliopathy, also I need to send them to Christoph because of this photoreceptor like a cilia. But now we focus on epithelium. Although the photoreceptor has a lot of mitochondria, the epithelium is more sensitive to inhibition of this Nobel Prize winning enzyme, protein hydroxylase. Because in the photoreceptor, although it has lots of mitochondria, it does have very little complex 4. So in the epithelium, you don't have complex 4 or the electron transport chain. Then you end up to have a lot of succinate. And succinate is also will inhibit this Nobel Prize in addition to this Nobel Prize winning enzyme. Not only does it require iron, protein hydroxylase, also requires succinate. So you don't have complex 4. Most of the succinate actually will become the electron acceptor instead of complex 4. If you don't have, you have a very high level of succinate, then this protein hydroxylase Nobel Prize enzyme will not work. If it doesn't work, then you're very high level of another Nobel Prize winning transcription factor. This is a hypoxia-inducible factor. So when hypoxia-inducible factor become very high, in fact, that's what photoreceptor like. Photoreceptor like to do Warburg glycolysis. It doesn't care if they have very high level of hypoxia-inducible factor. But the epithelium will die. Epithelium do not like to have hypoxia-inducible factor. So the hypothesis, at least it suggests that a lot of cells in the body have this kind of coupling, that the light-sensing neuron, in fact, Otto Warburg, usually I tell the audience that the TCA cycle is not used to sedate medical students. It does have some meaning, the TCA cycle. So the Otto Warburg described in retinal cells that people usually forget, and cancer cells. I guess that's why we have this cancer symposium across the street. Cancer cells, and also fetal cells that use as loss of lactate in the presence of oxygen, and that's what photoreceptor is supposed to do. So the photoreceptor makes lots of lactate, and lactate is used to become the carbon source for the epithelium. So photoreceptor like to do Warburg glycolysis, and then in aging, and also maybe in mutations, then the photoreceptor is not able to make lots of lactate. They become sick or they die. When photoreceptor do not make lactate, then the epithelium will switch carbon source. Epithelium is like a scavenger. If there's no lactate around, you switch carbon source. And you switch to glucose, because glucose is very close to the blood supply. The choroid has lots of glucose to support the photoreceptor. Usually the epithelium is like a conduit. Allow the glucose to go to feed the light-sensing neuron. So when the light-sensing neuron is not having died away, not making lactate, epithelium, instead of consuming lactate, will switch carbon source to glucose. When the epithelium switches carbon source to glucose, then it triggers a vicious cycle because glucose neurons, photoreceptor are neurons, and they cannot switch carbon source. Then it would start to die from starvation. So both the rod photoreceptor and also the cones eventually die from starvation because no glucose, because epithelium ate all the glucose. So this is some suggestion of why photoreceptor do the Warburg effect. So lactate is quite important to affect the level of when you do the Warburg effect, you instead of pyruvate going into mitochondria, you have make lots of lactate, and lactate is important in regenerating, taking care of the reductive stress, make NAD+. NAD+, in fact, is what Otto Warburg nominated for the second Nobel, and he didn't. He asked the Nobel Committee to ship him the plaque. He doesn't have time to go to Stockholm to collect the Nobel, so he did not get the Nobel for his second Nobel for discovering NAD+. So then you also, if you do lots of Warburg effect, you help the biomass. The epithelium has also the photoreceptor did all this membrane. Rhodopsin is a lot of lipids. So if you do the pentophosphation through the Warburg glycolysis, you can increase your biomass. And also the timing of making ATP is a lot faster through the TCA cycle than the oxidative phosphorylation. So this is a Nobel Prize enzyme, which is being discussed across the street, protein hydroxylase. It typically requires iron to hydroxylate the hypoxia-inducible factor. And then there's also, recently the HIF2 inhibitor is approved by FDA. So if you chelate iron, then protein hydroxylase will not work, or then the hypoxia-induced factor become pretty high. Okay? And I will show you some evidence that the epithelium, they typically like to do oxidative phosphorylation. Epithelium in aging, during aging, they go from oxidative phosphorylation to glycolysis. So this is taken from patients' cells in the dish. Patients' epithelial cell we make from stem cell. So usually they have a pretty low level of hypoxia-inducible factor 1. We add the iron chelator, you get a little bit higher. And then the hypoxia-inducible factor 2 usually is you don't see it at all. But you add the iron chelator, you get pretty high levels in the HIP2, even higher in the epithelial cell after iron chelator. And so if you in the iron chelator, but you can see that the cells are kind of sick and die. If you use alpha-ketoglutarate actually would activate. Succinate is an inhibitor, and alpha-ketoglutarate is an activator of this Nobel Prize enzyme, protein hydroxylase. Adding alpha-ketoglutarate, you can see that now all these dying cells will now survive with alpha-ketoglutarate. And then you can buy alpha. So patient kind of heard about this. You can buy alpha-ketoglutarate from GMC Nutrition, and patient took some, and it seemed to improve. This before he took alpha-ketoglutarate, his cone responses. After he took some alpha-ketoglutarate, that there maybe the cone response has some suggestion or improvement. So this is and this is not atypical for that one cell makes loss of lactate, photoreceptor make loss of lactate, epithelial cell eats the lactate. So one cell needs to make lactate, the other cell to eat it. This is also true in other systems. And in astrocytes tend to make lots of lactate, and neurons need to eat the lactate. So one cell needs to make it, the other cell needs to eat. And it's in the same thing in the Panna cell. Panna cell tend to also make lots of lactate, and then intestinal stem cells tend to eat those lactate. So there's it's not a balance. So if one cell then not balance it, one cell make it, the other cell eat it, and then it will lead to degeneration of aging. So now we come to this imprecision medicine concept. Actually, there's a tutorial. There's a tutorial from cell stem cell. They talk about maybe you need imprecision medicine because precision medicine may not be sustainable. This is the best example in cystic fibrosis. Vertex as a drug, highly effective for cystic fibrosis, only treat one mutation, arginine 117. And then the Vertex price it by usually when before the drug become available, the yearly cost for cystic fibrosis patient may be about $15,000 for taking care of pancreatitis, taking care of pneumonia. And they don't live very long. So the lifetime cost for a cystic fibrosis patient is about $300,000. And this is exactly the price the Vertex put for $300,000 for precision medicine. And then Lisa Hack and Randall here works on Stargardt and gene therapy. And it's estimated when Stargardt gets gene therapy approval, it costs $30 billion a year. And spinal muscular atrophy is about $100 billion. And then when the entire Medicare budget will be only $750 billion. So when gene therapy, precision medicine, very ready, when Stargardt, then maybe we have to close all the emergency room to support gene therapy. So that's why they talk about the stem cell, talk about imprecision medicine. So with someone common, if the Warburg effect is not good, you don't have good biosynthesis. One of the earliest defects, this is a retinal organoid, got from Peter Quinn, that you can see that there's lots of lipid in a normal, but in a patient, then there's not a lipid. So Warburg effect is not too good. Maybe there's a common pathway. So people study the rate of decline. Doesn't matter what, there are 80 different mutations and cause monogenic retinitis pigmentosa, but everything goes to the first order decay. So although the initial genetic damage may be different, you all went to first order decay. Maybe this first order decay is these problems in coupling or problem of lactate. So it's found in 2017 that by Joshua Rabinowitz and also Ralph DeBenedictis in humans and also in mice, most cells in the body, except the brain, except neurons, use lactate as the carbon source. Only neurons, you prefer glucose as the carbon source. So we think that maybe taking care of the lactate consumption and production, then maybe they will take care of many different genes. So we need to look at the Warburg the Warburg effect, PCA cycle. So this is not specific, right? So I show you in photoreceptor, one cell tend to be glycolytic, makes lots of lactate, and photoreceptor and astrocyte is a famous example. Inflammatory cells, M1 like macrophage, Panna cell also make lots of lactate. Stem cell makes lots of lactate. The niche of stem cells are typically fibroblasts, niche in cancer fibroblasts, and then your cancer cell also make lots of lactate. The niche needs to consume the lactate. The niche tend to be oxidative, and so as many of the Treg cells or the M0 like macrophage tend to be oxidative, and stem cells and cancer cells and Panna cell tend to be glycolytic. So this recycling, one cell make lactate, the other cell eat it, is not too new. So need a balance. So when we talk about Yamanaka factors, the 2012 Nobel Prize, Yamanaka factors essentially just to change a fibroblast. Fibroblasts have a lot of mitochondria. To change a fibroblast to stem cell, you're just increasing lactate production essentially and shut up mitochondria. So, and recently, Amazon, Altos, Juan Carlos, right? They are trying to partial reprogramming of to treat progeria, say. Partial reprogramming, you bust up the lactate to become stem cell. You partially make lactate, not completely. Then you suppress aging, and that's the platform for Amazon and Altos. So essentially, to make stem cells, I guess that's why I'm in Columbia Stem Cell Initiative. To make stem cell, you make lots of lactate. You need to shut up mitochondria, and then the niche needs to consume the lactate. And also, when a stem cell differentiate, then it also become more oxidative. But initially, what Yamanaka factor does is to shut up mitochondria in the fibroblasts, and then increase the lactate production. So, and also in aging, people looked at autopsy eyeballs in aging epithelium. They would go from young epithelium tend to be oxidative, aged epithelium then become glycolytic, and people have already noticed increase in hypoxia-inducible factor and decrease in the Nobel Prize enzyme in aging epithelium. So over aging epithelium would switch from oxidative to glycolytic. So and then we have interesting disorder. The patient's retina become like a dinosaur. They don't have any rods, only all cones, but eventually the cones will also die. And also many of the genes in retinitis pigmentosa are only rod specific, but why do the cones die? So we thought that maybe need a threshold level of lactate for the rods to survive. Also for the cones also. This is one of my very first patient that I started in my practice. The patient get lots of cuts in the legs. And so his friends at the restaurant thought that maybe he should get his eyes checked. The reason he get lots of cuts because all the cells died away outside, and then you only have a little bit in the center. So he bumps into things and got lots of cuts. The patient have a mutation of phosphodiesterase. And so in the mice, this is a patient with very salt and pepper retinopathy with a phosphodiesterase mutation. Mouse with phosphodiesterase mutation just look like people. That you can see that the vessels are attenuated, and also have a salt and pepper retinopathy. And then in the control mouse, you see the vessels are a lot more plump, just like the patients. So we can, the reason that a lot of the gene therapy trials are done in the retina, because you can count them. You can count this nuclei with optical imaging. You can do an electric response in the retina. So we have a mouse model of phosphodiesterase. Over time, you can see that the nuclei kind of disappear into a single row over time. And the electric response is diminished. So this become an outcome measurement. So we can do gene therapy. In gene therapy, we try to increase lactate consumption or lactate production by gene therapy. So we can engineer the epithelium to be more robust oxidatively, so you can consume lots of lactate. We did not treat the primary defect. The primary defect in these mice have a phosphodiesterase defect. We did not apply the cDNA. We can just improve lactate consumption of the epithelium. We did not do anything to treat the disease photoreceptor, but just increasing lactate consumption, we can have more cells survive compared to control. And this is with gene therapy is focal injection. Focal injection just around the yellow area, far away from the injection site. There's no effect. So we get a pretty good effect locally just by improving lactate consumption, epithelium, and the electric response also improved in the patient, in the mice. So the name of the condition is called retinitis pigmentosa, with lots of pigment. Improving lactate production, you can see that in the treated eye, even the pigment disappear. And we count the number of cones. So you just improving lactate epithelium consumption at epithelium. We did not do anything to the photoreceptors. We did not treat it with the cDNA. Then we can see that the number of cone cells density also improved. These are some of the work done by a new assistant professor Wang Nankai here. So we need just to show you the improving lactate consumption. We can slow degeneration. So there's another way, improving lactate conduction, removing ablating hypoxia-induced factor in the epithelium. We can also decreasing glycolysis in epithelium. We can also see that the nuclei of the also survive compared to control, and the electric response in the retina is also better by just improving lactate consumption, decreasing glycolysis in the epithelium. The number of the dots, you can see the cone cells also improve compared to control. And also we can do other means using microRNA. If we have the microRNA that controls mitochondria, you ablate this microRNA, you can see that the oxidative phosphorylation, PGC1 alpha, improved in the epithelium. And we can see some transient improvement in the nuclei, the survival. So we did not treat it with the cDNA. We just improved oxidative phosphorylation, decreasing glycolysis using microRNA ablation in the epithelium. We did not treat the photoreceptor, but you can see improvement in photoreceptor survival. You can see that the glycolysis went down in the epithelium. So you have less glycolysis in the epithelium, then you can able to consume more lactate. Lactate dehydrogenase level in the treated eye here, it has a lower level of a lactate dehydrogenase compared to control, accounting for the improvement in cell survival here. A little more nuclei, just decreasing lactate dehydrogenase in the epithelium using microRNA manipulation. So now we show you the example of eating more lactate. I'm also a member of the Institute of Human Nutrition, so you need to eat all this lactate. Then you need to now produce the lactate. So you will remove SIRT6, you increase this Nobel Prize factor, 2019 Nobel Prize factor, hypoxia-induced factor, improving glucose transporter. And also you can see that in the control, this photoreceptor nuclei eventually disappear, the single row, the yellow bar, this photoreceptor nuclei. If you improve lactate production, then you can see that the nuclei also improve. So more lactate can feed the epithelium. So you can see that by C13 tracing with the help of Jim Hurley and Jin Hai, they can see that the pyruvate, lactate, all the typical TCA cycle metabolites increases as predicted. There are other ways to improve lactate. We now knock out the Nobel Prize winning enzyme. Then we knock out this Nobel Prize winning protein hydroxylase. Again, hypoxia-induced factor increases the phosphofructokinase level, and then the cells survive much better. You can see that the nuclei also improve. The electric response in the retina in the red is after knocking out this Nobel Prize winning enzyme in the photoreceptor. We did not treat it with the cDNA, just removing this Nobel Prize winning enzyme. Then we can see more cells survive. Then with this C13 tracing, as expected, then the nothing enters, less enter the mitochondria. The dotted line as a control, so we don't see a lot of metabolite like citrate going into the mitochondria. We have more TCA cycle, the dotted one in the cytoplasm. So once you remove this Nobel Prize winning enzyme, then glucose is not able to enter the metabolites in the mitochondria and strengthen the TCA cycle. So we also get more cones, improving compared to control. So this is the one side we will remove improving lactate production. You also see more cones. This is a high MAC. This is control. Few dots. This you see more dots after improving lactate production. So in the mice, we can also do focal injection. So we can only GFP marker to show you that we can see a very focal transduction. But we get some global, because lactate is diffusible, so we get some global responses. So then in this case, we use our recessive model, the phosphodiesterase, the same enzyme complex as a patient that works in the restaurant per se. Then you can see that this is a local area of transduction with the guide RNA, but we can see pretty global improvement, both the electric responses in the retina on the top trace. And then the cells also, compared to control and injected eye, you get more dots, the rows of nuclei. The lipid, the outer segment, is also better than control. This is a focal injection. So in the spirit of imprecision medicine, we treated both this recessive model and treated a dominant model. This is one of my patients, the grandmother. You can see that all these cells died away except the little center. And this is a grandchild with dominant rhodopsin mutation. So we also worked. We did not say treat rhodopsin. We just improving lactate production very focally, only a small area. And then electric response also improved with a focal ablation of this Nobel Prize winning enzyme. And the cells, this is a slow degeneration. So you can see that the number of cells and also have more compared to the AAV blank control. So I show you that the strategy of improving lactate consumption and also lactate production, we may be able to treat many different, all these 80 different mutations. So I show you some of the strategy that how we apply genome surgery. First, it's a precisely a nucleotide at a time at a single residue. I'll show you how to use it to solve genome-wide association studies controversy. We show that using single nucleotide variant to map the chromosome and ablate anything linked to the chromosome. So we now can treat hopefully all the 150 mutations in a less precise way. And then maybe this is precision targeting of the pathway. We just talk about lactate, but this may hopefully is a precise pathway, but then we can be also targeting entire pathway to treat mutations in 80 different genes and call this monogenic disorder. So this is last week I was at NIH, and I hope that maybe the US system of should be same as in Germany. This is the extent of Otto Warburg's grant application is in German, which I cannot read, but translated. I require 10,000 marks. This is the extent of Otto Warburg's grant application. So I think this the Krebs cycle, the TCA cycle is a very fruitful area. So this is maybe this is a kind of application that you can send to NIH. And then the people, and Otto Warburg described the Warburg effect in lactate production in the presence of oxygen in cancer cells and retina cells. Retina cells looks like cancer cells. It express very high level of pyruvate kinase M2. Retinal cells and cancer cells express high level of hexokinase. That's like cancer cells, LGR5 positive, and that's like cancer cell has lots of succinate in photoreceptors. So we talk about a lot of sequencing genome, and knowing the genes, precision medicine, and knowing what the paint is, yellow paint and blue paint. Now the people like RNA-seq, so like looking at the brush to see where the RNA or proteomics, transcriptome, single cell sequencing. But I think if anybody recognizes this a statue in the Metropolitan of this figure. Anybody recognize this figure? And in the Metropolitan, in the American Wing, it's actually a very good landmark. It tells people you want to meet at this statue in Metropolitan of this figure. This is the only survivor of Pompeii. and she's blind. So this is the blind flower girl of Pompeii. So if you need to know the whole picture, I think you need to also precisely know the metabolites, not just the sequence of DNA. So then I think all the mice, they volunteered their lives, and they C13 tracing with the help of Jim Hurley, Dan Haas, and Jin Hai, and to age the retina in the dish, we get help from Janet Sparrow. Dita taught me how to make iPS cells. My classmate in graduate school, Chen Sing Lin Victor, taught me how to do embryonic stem cell and do all the blastocyst injection. And I'm happy to answer any questions.
It's a very basic question, I guess. So you said Warburg, preferentially observing Peters and cancer cells, tumor cells, and
photoreceptor retina. So that's what that was described around the Second World War. So,
yeah, that those cells, retinal cells, don't divide as compared to.
but they have membrane. Retinal cells need membrane. So because the rhodop very membrane rich for this cilia, and in mice, everything is turned over in 10 days. They need to make membrane. So cancer cells, part of the suggestion why cancer cell needs this Warburg effect. In addition to, you make lots of lactate. When pyruvate becomes lactate, you make a lot of NAD+. So cancer cell needs a lot of NAD+, which is what Otto Warburg discovered NAD, although he didn't get the Nobel Prize because he didn't want to show up in Stockholm for the NAD. So Otto Warburg described NAD. So cancer cells and photoreceptor need lots of NAD+. And also, they call cancer cells this reductive stress. If you don't have enough NAD+, you have this reductive stress. And also, in addition to, if most of the pyruvate don't go to the mitochondria, you also start to feed into the pentophosphate shunt. And most of the lipids are made in pentophosphate shunt. Cancer cells, stem cells divide rapidly because of the membrane, and photoreceptor need to renew the membrane. So rhodopsin, which I described to you, is on a long distance membrane. So photoreceptor, although they don't divide, but they need a membrane. So you need membrane, you need to use a pentophosphate shunt, and that's why you use a Warburg effect.
That's not like what you told us, that you take different, including RNA and some of the TVs, to all these different genes to increase the lactate, then improve the pathology. That's very interesting. What's there's no mechanistic connection?
So people think that the photoreceptor die from starvation of glucose. So the hypothesis is that you want, I guess that's why I'm also a member of Institute of Human Nutrition. You want the photoreceptor to have glucose, and you want the epithelium cell not to use glucose, although it sits very close to the blood supply. You want the epithelium cell, like any cell. So Josh Rabinowitz at Princeton showed that in humans that pancreas and lung cells actually wants lactate as a carbon source. Most cells in the body use lactate as the carbon source to feed the mitochondria. Neurons exception. Neurons, photoreceptor are neurons. Neurons use glucose. So usually, in the Warburg effect is that the photoreceptor makes lots of lactate to feed the epithelium. But when photoreceptor becomes sick, it cannot feed the epithelium. Then the epithelium switch carbon source. Photoreceptor, because it's neuron, it's not flexible. And versus so photoreceptor is not flexible, then you can only use glucose. But epithelium is very flexible. If there's no lactate, you just switch to glucose. Especially glucose is so close to the choroid. Then it trigger a vicious cycle. a vicious cycle, and then the rods start to have not enough glucose. People show that glucose are trapped in the epithelium, in aging retina, say, and then the cones eventually also because die from glucose starvation, because cone photoreceptor and rod photoreceptor cannot switch carbon source, but it's just the epithelium can switch, just like any cell, like the pancreas and the lung.
So regardless of mutation, different genes that cause this retinopathy, maybe increasing lactate feeding, maybe that helps.
So far, I think they work. So either you can,
Yeah.
but I don't know. You can. It's I don't. Kind of difficult to envision to put a catheter in the for a catheter to give a lactate, but I think we try to do it genetically by manipulating this Nobel Prize winning enzyme that you hear across the street.
So even someone who has developmental defects, this bone receptively, you know, or this the whole retinal degeneration problem, then
yeah. So people observed that that actually was a review in the annual review of medicine, or annual review of genetics, that Rod McInnes and Alan Wright, they look at different rates of degeneration in mice, and also different genes, and also combined with Sam Jacobson looking at it in humans. So it doesn't matter what gene it is. It always goes to a first order decay. So initially, different genes determine the age of onset, but then the rate of decay is the first order decay, always the same. So they were arguing that there's failure of mitochondria in the annual review medicine paper, and said that the mitochondrial failure account for the first order decay. So it's kind of related, right? Mitochondria. If you don't feed the mitochondria of the epithelium with lots of lactate, then the epithelium starts to steal glucose. At least that seems to suggest a common, maybe a precision pathway. And it's probably the same in other tissues. Neuron, maybe in Lou Gehrig's motor neuron versus the astrocyte, this kind of lactate cross-feeding. At least it works in stem cells. Stem cell, the niche stem cell is glycolytic, like cancer cells, photoreceptor, and the niche tend to be oxidative.
I just want to make sure that our virtual attendees have an opportunity to ask questions as well. Are there any questions online? I don't see anything in the chat.
Yeah, any question from the chat audience? Other than me can make a comment, I didn't see any other.
Great, thank you so much for a fascinating talk.